Again, the Let's Talk Forensic with Pastor Dan. We're going to do one more day of notes. We're going to, again, looking at blood analysis, and as we defined it before, blood analysis is defined as study of blood and parts of the blood that can be determined to determine differences in an individual or identify clues in a possible crime. Now, what we're looking at today is something called blood splatter analysis. Now, this has gotten a really huge area, and you can actually get certification to be an expert in blood splatter analysis. All right, so what exactly is blood splatter analysis? Blood splatter analysis is a means of determining clues as to result and cause of a crime by looking at the patterns of blood left behind. So the blood that is present at a crime scene, we look at and be able to identify key evidence that we can point back to the victim, the perpetrator, and the crime that was committed. All right, now, the history of it, so we understand how this came in. The earliest reference to blood splatter was in 1894. In 1939, the first actual research into the technique began, so we can begin to be consistent in the process. In 1955, the first time it was ever used in a trial in a murder case. And then 1971 is when it was used as a tool in a modern forensic analysis. So it began the first time it became commonplace and began to be used in a much more rapid method. Now, blood itself is composed of three main parts. First, it's composed of red cells, blood cells, which are the hemoglobin, the antigen, and that's where your DNA is. It's in the red blood cells. Second is the white blood cells. That's your antibodies that fight infection. And the third is plasma. That's a saline nature that makes up all blood. Now, reasons that blood patterns are found. First of all, it's something called cohesive. That blood itself is cohesive. What does cohesive mean? Cohesive, the tendency of a liquid to stick to itself. So like mercury, it balls up. And it's also absorbent. So that means it can be pulled into other mediums. So if it hits something, it can actually blow up and be a drop. And then if there's a material there that is absorbent at all, it will be pulled at itself. Now, cohesive is different from adhesive. Cohesive, like mercury, balls up. Adhesive, like water, pushes on the edges and raises on the lips, which is the tendency of liquid to stick to other items. All right. Now, in terms of blood itself, we can also determine the distance blood falls. We do that by looking at the size. The blood falls as a small sphere. It's a small sphere when it falls and leaves a droplet. Now, as it drops, the farther it drops, the bigger that droplet's going to get because of the fact that the speed is going to increase. Types of blood spatter. There are 5 types of blood spatters. Pooling. So if you get stabbed and then you're lying down, that blood flows out, that's a pool. Okay? Second, splatter. So like if you get a nosebleed and it's bleeding straight down, that's gravitational. Gravity's pulling it down. Individual round circles, gravitational blood splatter. Directional, if you have a nosebleed and you start running, as you run, that splatter is going to get elongated and be able to identify a specific direction. Cast off, if you're getting beat with a baseball bat, as the person pulls back, the blood flings off. As he hits you, the blood is cast off. So all those things are called cast off. That's 4. And the last one is called blood spray. This is really bad. Hits an artery and shoots out with velocity because of the heartbeat. So those are the 5 different types of blood splatter that are possible. Now, blood can be used to determine points of origin using 3 main things. First of all, following the blood trail, using what's called lines of convergence, and lastly, identifying what is called a void. Let's go through each of these. Okay? Determine the direction of the blood. A directional blood stain will end up being pointed as the person's running, it hits and then gets elongated. And the point of it will show the direction that the person's traveling. So you literally can, if you've ever hunted, you know this, because as you hunt, the blood cuts in there, and if the animal's running, you can follow direction by which way it's pointing. Second is called the direction of blood through line of convergence. What does that mean? So we take all these lines of convergence, all these directional gravitational spreads, and we pull them back to a single point, and then from there we can identify where the victim was when they originally attacked, and all those points come together, and that splatter pattern is what we call the points of convergence. Lastly is a void. A void is an absence of blood that identifies the presence of a person or object was present. So someone's getting beaten, there's blood everywhere, and when the person comes in, there's big open spots, something like this. So this would be the crime scene. There is a void here, could be a person, and then we have a void right over there. Maybe a small item the person picked up, so we can identify based on size, height, things along those lines. All right, now to determine size from a blood space, we can identify that the height from which the blood drops. Now there is a maximum height. We can also identify the type that was used, type of weapon, if it's a knife, gun, things like that, part of the body affected. Okay, the faster the drop, the larger the size, the diameter, the size. Now, this will continue, okay, until you reach a maximum of 7 feet. At 7 feet, we hit what's called terminal velocity. It won't increase in speed up anymore, and so blood will just hit that same size. So up to 7 feet, that's a maximum speed available, okay? All right, the effect on surface. If we hit something that's smooth, they get a nice round spherical drop. If, on the other hand, you hit a rough surface, it's going to be splattered out, so that will also affect how it appears. All right, that's it. Hopefully it's not too bad. Take care. God bless.